Salutations! You better hold your breath. We're going on a deep dive into advanced methods in Multivariate Analysis 3, Deep Dive. I'm Dr. Mark Williamson for DaCCoTA at UND. Last time, we covered Mancoba, clustering, and recursive partitioning in more detail. Today, referencing our 6-group schema, we'll cover canonical analysis and time-based methods in much more detail and examples. So let's review the basics. These are our 4 canonical analyses, which are all essentially looking at comparing one matrix to another. Now, correlation analysis is symmetric, which means we don't have any assumptions about like causality, like X is causing Y's and so forth, so we usually call it a U and a V matrix. Whereas corresponding redundancy are asymmetric, as well as linear, where there's a specific Y variable or Y matrix of variables and an X matrix of variables. These two differ because of how they estimate differences and sort of some of their other assumptions, but that's a big one. Where linear discriminant analysis, Y is not so much a matrix but a class of objects. So it's like multinomial regression, but there's a dimensional reduction in sort of the X matrix. So I showed this in our first slide. This is the little thing I created to sort of parse out which one of these to use with your data. And again, the big thing with canonical analysis is it's a combination of ordination and regression together in one sort of analyses. Now, turning to time-based, we have vector autoregression and principal response curves. Vector autoregression has looking at seeing how 2 different time series may be related to another, whereas principal response curve is more like a repeated measures type analysis, but for multiple Y variables. And it's usually plotted something like this, where here's sort of like our Y variables, this is our over time, and then the different curves are different treatments we're interested in. Okay, so let's get more into some of the details, especially of canonical analysis here. So what the canonical means is, from the dictionary, it's confirming to a general rule or acceptable procedure. So you think of a canonical analysis as more like a generalized form that's more flexible to fit types of data. So if, like, for example, there's like general linear models which have certain assumptions, but then there's generalized linear models which have less assumptions. They're more complicated and complex to use, but they're more flexible. That's kind of what we see here with canonical. They're more flexible, but are more difficult to interpret sometimes. Another good definition is that canonical analysis as a whole, they're multivariate techniques which confirm the relation between a group of variables in a data set where, like, there's 2 groups, X and Y, U and V, whatever you want to say, and you want to find a relationship between the 2 of them, not too surprising or confusing on sort of a broad scope level. But let's dig down a little bit more into like what makes different types different. So again, I talked to you how kind of close corresponding analysis was with redundancy. So let's kind of parse that out a bit. So both do stem from ecological resources, where you typically are looking at a bunch of Y variables, like abundance data of species, and then a bunch of X variables, a lot of times like environmental variance, like pH, temperature, whatever. And so what kind of makes these 2 different? This is some sources I got. So RDA is apparently redundancy analysis. So I'll just do RDA here and CCA, just so we have the acronyms right. Short gradients, though that's not clear that actually matters. Another helpful bit here is that CTA is a little bit better for if you have a strong a priori hypothesis on constraints. And so redundancy may be more better for data analysis, where CCA is like, I have specific hypothesis I want to test, and I'm not going to throw everything at it. I'm going to have a more constrained set of variables. That's good for here. And again, CCA focuses more on species composition or relative abundance, whereas RDA is better at sort of looking at a gradient along all species. So again, These are more ecological considerations. So when we look at examples, we'll try to sort of translate it into more of our data here. So keep those sorts in mind. Now, looking at the time-based part of it, what makes vector autoregression different from principal response curves? Well, vector autoregressive is usually interested in predicting multiple time series over a single model. And so this is an extensive univariate where apparently, so don't take my word for it, take this resource's word for it, that principal response curves is a special case of redundancy analysis. And so you have treatment's a factor, and then a factor of time points in repeated observations, and this is what the model kind of looks like. So maybe what that means more on a non-technical level is vector autoregression is usually pretty common in economics. Like you're looking at the change in like stock price of one commodity versus another one, where principal response curves is more common in ecology, where you're looking at treatments over time. So hopefully that's at least a bit helpful. So let's jump into our step-by-step example 1. So we're going to use canonical analysis using the CARS 93 data set. So we use MT CARS in the last time, so we're going to continue our car heavy theme. So the 3 questions we're going to ask is, how do car variables and engine variables compare to one another? So this is going to be one matrix of variables and another matrix of variables. We're also going to ask, can car variables, so this is our X sort of matrix, predict the type of drivetrain? So this is a class. Y is a class. Type of drivetrain. There's 3 different types. And then finally, we'll look at how do car count variables and metric variables compare to one another. We'll use both canonical correspondence and redundancy since they're very similar. Once again, we'll be using R exclusively for this because R can do everything. If not perfectly, it can do everything. So we need a whole bunch of libraries here, and then this is our data set. I'm not going to print out the head because that's pretty wordy, but I will just show the names. You can see here that CARS 93 has quite a few variables of different types that we can use. That's why I picked it. So let's get going with canonical correlation analyses. So we want to set up our variable sets. So remember, we're going to essentially have a U matrix and look at that compared to a V matrix, because this is symmetric. We don't have any sort of a priori think of Y and X, whatever. So we're going to create a data frame for fuel, which include miles per gallon, fuel tank capacity, and weight. And then one called an engine data set, which has engine size, RPM, and then revs per mile. So some assumptions, we're going to test for normality. So we're going to look at some histograms, and all these variables, they're going to be close to normal enough that I'm going to say that's fine. Okay, they're not terrible. And then let's look at some variable sets here. This is nice to kind of look at correlations. So this is our fuel variables. We have miles per gallon compared to fuel tank, miles per gallon, and weight, and then fuel tank capacity and weight. And you see that, yeah, there seems to be pretty strong correlation between all of them. Similar here for now, engine size, RPM, and revs per mile. So that's just some data exploration here. All right, let's get into our correlations. So we're going to start with some correlations at and between variable sets here. So we can use MatCore. So this is just a bunch of correlations. We can do this with ProcCore, or not Proc, just the function core. So there's correlation between X, Y, and then X and Y. So it gives us things. We've already seen some of this sort of in the visual form. Weight is highly correlated with miles per gallon, unsurprisingly, and so on and so forth. So let's dig more into our canonical correlation. We're going to use CC, save it as this object CC1. And we can pull out correlations by going here. And so we have 3 correlations, and they are popped up here from descending order. We can get raw canonical coefficients here, clean up here, and that's what we get. And essentially this kind of read like normal sort of correlation for this first canonical essentially axes. This is how these X variables are related, and these are Y. R calls them X and Y. I would probably call them U and V just to be consistent. For example, here engine size is very strongly correlated with this first one, whereas these other ones really aren't, in fact, really at all. Whereas, like, for example, fuel tank capacity is not really strongly correlated with this first axis, but it's much more for the second one. So we get kind of those sort of things. And then we can get these canonical loadings. Again, we're going to kind of clean this up. There. Okay, that's better. Now let's well, mark it up again. And so these are all sort of our loadings, and they're all sort of a type of latent variable again. That these latent variables are trying to capture sort of this variability of all these variables. Anyways, so we can do a significance test for canonical dimensions, because like we have 3 here, but maybe we don't need 3. So we're gonna have the correlations. We're going to keep that in row, and we're going to add, just make these variables like these numbers, like how many variables have in the x and the y and so forth. And then we're going to do a series of tests. There's a variety, so I'm using both a Wilks, a Hotelling, and a Pillai. And then we can get significance of our first, second, and third canonical axes. And it turns out the big kind of thing here is only the first is significant in all 3. The Pillai, the second 2 are significant, but they aren't in this one. So I'm going to go with the fact that only the first sort of correlation axis really is significant. And then we can get standardized canonical coefficients. Again, this is sort of the same sort of thing we see here of having how well they're correlated to these different loadings. And this is the sort of summary you could kind of write up for what we found here. So these were our tests for canonical dimensions. It turns out I'm just using, I think, the Hotellings. Only the first dimension was significant. And then for all our significant dimensions, we're going to sort of have our standardized canonical coefficients. So dimension 1 here. We can see here how we can interpret things where like weight was strongly positively associated of our fuel variable with dimension 1. These ones are less so. Engine size was strongly correlated. RPM really wasn't. And then revs per mile was actually negatively correlated. So this sort of steps that I kind of went through on the CARS 93, it's based on this reference here, which used a different data set, but the same sort of method. So you can look at that for another same approach in a different data set. So really great stuff. Okay, let's switch over to linear discriminant analysis. So now we're going to set up a different data set. So we're going to have our CARS, we're going to call LD for linear discriminated. Now we want to keep drivetrain in here because that's going to be our class Y variable, and then all these other X variables that we have in here. Linear discriminant analysis, it's recommended to scale it, so we're going to scale it. And then we have a mean essentially of 0. These are all essentially 0, and then a standard deviation of 1. Then we can essentially linear discriminant. You typically sort of like that machine learning thing where you separate some into training and testing. We're going to set a seed. So if you want to reproduce it, you use the seed 1. And then so 70% will be of our data will be a training set, while 30% is testing. So if we keep going here, we have our model. So drivetrain is a Y variable, and then this tilde and then the dot is sort of shorthand for every other variable that was in that data set. That's going to be our X's. And so when we call LDA1, so here's our model, we get prior probabilities, group means, coefficient, and then this proportionate trace. And then actually you can make predictions. So if I can save this as predicted and using our test data, because we trained our data with this, we can test our data. And then we can, there's all sorts of things we can, turns out there's a class, posterior, and X in this predicted data set or output. So we can look at the different classes, which is four-wheel drive, front and rear. That's the drivetrain. We can show our posterior values, and then find our predicted values. So you think of posterior, predicted values. That's Bayesian terminology. So I'm sure what's going on under the hood is Bayesian stuff, which I'm again going to sort of hand my way that and be like, well, we don't need to know that much about this, only like the outcomes. Anyways, and then we could find accuracy of model. So it turns out if we do predicted class and then tested our trend, it turns out our model is about 80% accurate using all those X variables to predict whether a car will be four-wheel drive, front, or rear wheel. So not perfect, but maybe be good enough for our methods here. You can see how linear discriminant analysis can be pretty useful, kind of analogous to some of those tree-based methods we saw before. All right, hey, let's visualize the results of our linear discriminant analyses. So we'll just set this up with ggplot. We get this outcome here, and we can see, why maybe we only have an 80% success rate. Looks like if against our ordination, if you're kind of in between the two here, you're most likely to be front-wheel drive, but it's not perfect. Here's a four-wheel drive. Four-wheel drive is sort of interlaced between them. They seem to be the hardest to predict. And then rear is kind of also messing up in here. So you see how kind of similar to our sort of tree-based methods we saw earlier. Now, if you want to see, again, this is a great resource here, this site. It's the same. This is where I took sort of this method. They use a different data set. If you want to see this sort of same walkthrough of the methods with a different data set, you can go to there. Okay, so let's take a look at canonical correspondence and redundancy analysis. So this is just some texture I had here explaining some more things. It turns out with the what we're going to use here, you can add actually three different kind of matrices, X, Y, and Z. Z is called a conditioning matrix. If you include that, that's actually what's called partial correspondence or partial redundancy analysis. And if you have just X and Y, which we'll be doing now, we'll just regular old canonical or redundancy. And of course, if you just have one, like an X-axis, that's principal components analysis. Okay, so we'll create data frames. So now we're doing a different kind of data frame. We're trying to sort of model the ecological setup where we're going to have like these sort of count data, almost like abundance data for cars. So like cylinders, number of cylinders, number of passengers, and then fuel tank capacity, all kind of more county things. And then more just other metrics like engine size, RPM, and so forth. We're also going to make sure cylinders is an integer, and then just look at the structure. So we have two different data sets. These are going to be sort of our Y variables, and these are going to be our X. This is just okay. So let's start here with a basic CCA with all variables. So this is going to be our X and our Y variable here, and this is the output of just calling this. And then if we want to plot it, we can do a biplot here. And then what we kind of see here is our X variables, our car metrics, are these blue. These are our canonical correspondence axes, and then actually the red here, those are going to be our Y, our car counts. We can see here that kind of pretty grouped together here that this might not be the best method. So one thing I'm going to do here is make a little bit more nuanced formula because CCA is kind of better for that, more a priori sort of hypothesis we want to test. So I'm going to, instead of looking at everything and just one by one, I'm going to have engine size, RPM, and revs per mile, including those interactions, and then price by itself. And go out here, and this is what we get here. Not really much better with our car metrics. They're still kind of scrunched up, but our different X's sort of change. So now let's turn to redundancy analysis, but first here, let's clean this up a bit. So if we do the redundancy, exact same code as the first one here, except for we're going to use RDA instead of CCA. And when we call that, we get these sort of eigenvalues, and stuff. But here's the biplot, and we can actually see here that these are split out a little bit more. So this is maybe a better approach given our data set. Though some of this interpretation here, I'm not going to go really into too much. It kind of takes a lot of detail to sort of piece out what it's actually trying to tell us here. Though, if you want a really nice, again, this is where I took this approach. It's using a different data set. You can see the same method, different data set. So that goes into much more detail about some of the interpretations. So there we go. Okay, so let's change gears a little bit and ditch our CARS data set and try a different example with our step-by-step example 2. We're going to use time-based methods. I couldn't really find a good just base data set in R, so I'm going to use the simulated data and the PowerFOS data set based on the references that I'll show at the end of these things. So we're going to ask the question, how do two time series relate to one another using vector autoregression? And then how do doses of pesticides, so that's the PowerFOS, affect invertebrate abundances across time? So that's with principal response curves. All right, let's get fixing with vector autoregression. So we'll start out here with some of these just set up. So set a seed because we're doing some auto generation. Essentially, we're just going to simulate data, time series observations, endogenous variables, legs, and then generate these coefficient matrices, how they relate to each other. Coefficient matrix of like 1, 2, and 3. And you can really just put any sort of variable between 0 and 1. I did different things here than the example I took this from. It gives same sort of general outcome. And then the A is going to be our companion form for the coefficient matrices. So then we'll use this sort of information and a for loop to generate our series, and then convert time series, rename them, and then we'll plot these here. And really this is all just to simulate data of series. We have time series 1, time series 2, and they have some sort of relationship to one another because we gave them coefficient matrices. All right, so let's continue with that. So we can estimate the model, and with these 3 steps of VAR is vector autoregression, and this is the outcome we get here. I'm not going to really focus too much on it, but we can see here that there's p-values. You can see what's significant and what is not. And we also have covariance matrix of residuals. So going on here, so we can actually take our true values that we had of that coefficient matrix, and then we can extract the coefficients we got from running our model and look at the estimated ones. And we can see here that, oh yeah, these are actually pretty close. Normally we don't have this information because we didn't simulate the data and set things ourselves. So this usually this is what we're trying to sort of get out here. And then finally, one thing you can do with turns out you can do with vector autoregression is you can look at impulse response. So the effect of a shock from like series 1 of time series on another one. So we have like our impulse and then our response. So series 1, its effect on series 2, and then for a certain period of time again, it can look like this. We can see that, oh, there's initially kind of a shock, but then it kind of sort of stabilizes after time iterations. You can also have the cumulative. So that's cumulative goes true, and we can see here that sort of over time it ends up being positive, slightly positive in the end here. So again, you can turn to this for much more detail. This is definitely used much more in economics, so it's sort of less relevant and more in my biomedical field. So definitely go to them for more detail and why you need some of these things here. This is just I'm sort of skimming the surface, just how you roughly do it and get it done, and then interpreting results. There's a little more nuance to that. I'll leave to better heads. Let's switch over to the ecological side of the street by using principal response curves. So we're going to be using this data set of chloropyrus experiment. So there's a pesticides treatment in ditches followed over 4 weeks. And so like the outcome, like the Y variables they were looking at is like these different invertebrate abundances. So there's all sorts of invertebrates and they're relative, or maybe not relative, but just counted abundance. So we got week, dose, and ditch. And then in our principal response curve function, we're going to have paraphos. That's our response variable. So it's a huge, it's like 100 plus variables of abundances. Our dose, so that's our treatment variable, and then week, which is our time variable. We need a response, a treatment, and a time, which we have here. And then I'm just going to show a summary of some of these things. This is our formula, but I just want to show like, so this is all sorts of different species and it's their score. A lot going on here, which is why it's often plotted, which we'll do next year. But you can print that out if you want here. So we're going to plot, but we're only going to plot those that have a log abundance greater than 100. So because like there's, like I said, over 100, so we just want to kind of whittle it down to the more relevant ones. And here we get here. So we have our doses. So those are our treatments, those lines over time, and then these are all our species abundances. So this, not the most helpful. It's pretty busy trying to interpret this stuff. So what I did was I reduced things out here and only included the first 10 species, ran this again, and then plotted it, and here we go. Here we can see that most seem to respond negatively, but one seemed to at least respond positively. That's what I interpreted from. In fact, what I kind of did here is look at invisible abundances, just sort of get a feel for like what this is trying to summarize. So this is not to go into too much detail here. These are like the first 5 of those species, and then plots. You can see here that this is the change like abundance over time by dose, and we see most kind of see sort of like a dip, but it's sort of kind of all over the place. You see that there's a lot of different responses, so it's nice to be able to kind of summarize it. Anyways. Again, this is sort of complicated. It takes a lot to like piece apart, but this can be a pretty powerful technique to look at sort of this repeated measures with lots of live variables. Again, I will point you to cooler and better heads than I. And something interesting here is, I don't know why, my initial plot is a lot flatter than what they get here, and I'm not sure what the difference is. So they have a little bit more interpretable plot, which is always helpful. So anyways, I've combined steps 1 and 2 examples into just one kind of assessment here. It's not going to be in any of the role of coding or interpretation because that's pretty complicated. It's just going to be sort of high level sort of understanding of some of these tests. So you can go and take that here. Links are in the description below. All right, some caveats and concerns here, they are many. So just general, any of these tests, you really need to be able to slowly walk through and interpret the fire hose of outputs a lot of these things have. Therefore, sometimes, depending on what you want to do, a simple analysis might be better, it might be more interpretable. But if your data and your sort of hypotheses have need of these are definitely elegant and complicated ways to struggle with the vast, complicated world that we live in. Anyways, so more particularly canonical correlation analysis is some things here is there are an assumption of normality for both sets of variables, so that needs to be remembered in your analysis. And then the note here is it's bad for small sample sizes, so you want to have a lot of data for that. Now, canonical correspondence analysis, overfitting can be a danger, and the interpretation really depends on that good set of your explanatory, your X variables. And one of the things here is what's kind of going on here is you kind of have these sort of latent factor analysis sort of going on here, and sometimes that's not always helpful because you actually might care more about measurable X variables and Y variables rather than sort of like an underlying sort of combined latent factor. So, and then for vector autoregression, just a reminder, different number of legs can be set. And then principal response curve is sometimes graphs are hard to interpret with too many variables, so you might want to think about your life before you throw in like 100 plus Y variables with insect abundance or whatnot. So just a thought. Okay, so let's peek into some real world examples here. So we got canonical correlation analysis. This is, again, like I did for module 2, these are all from mostly from +1 because I could look at the methods. And so this looks at omics measurements and then clinical phenotypes. So that's the U and that's the V matrix. And what they're able to do for there's tuberculosis and malaria, they're able to sort of have a proteomic profile and a clinical profile and actually kind of separate groups, a little bit less so with the malaria, but it's kind of interesting. All right, with linear discriminant analysis, this is with some paleontology, some cool stuff here. They have the species, Lycophan, hutchinsoni, and what they are able to do is use linear discriminant analysis with these different dietary, between 3 dietary groups, and they're able to find that it's most likely to predict that it's going to be a a mesocarnivorous diet, that the open circles are going to be carnivores, and this is it itself, and the ones that it's most closely related to all were carnivores, so it's most likely a carnivore. So kind of interesting stuff here. Canonical correlation of this population structure. So we have gastropod species, those are our Y matrix, abundance data, so that's our Y matrix, and then our environmental data is sort of our X matrix, and this is what they get here. Again, we see these are the different species, those Ys, and then our X's. You can see here, like for example, salinity seems to be most associated with these species, where death is more with that one, and so forth. Redundancy, similar kind of thing here, though we have a soilmite meta-community, and I think, but I think their X and Y matrices are spatial and environmental variables. And they just had a table rather than a graph, and what they found here is that PC1 and PC2, their ordination axes, were influential. So that soilmite community were influenced by PC1 and PC3 in 2012, but that kind of shifted a little bit, where like PC4 was also significant in 2013. What that really kind of means at a deeper level, I'm not sure, but you can do that. So, vector autoregression here. So this I had a hard time finding a clear one here, but essentially they looked at this partial range of causality, which is based on multivariate vector autoregression. They're trying to look at how to sort of model, I think, orienting of attention over these different domains of sort of the brain over time. So again, I'm not really sure how to interpret this, but they used vector autoregression to do interesting, complicated things. Finally, we have some principal response curves. This is sort of the classical sort of paper that they came up with this and doing this. So we got this here. You can see here a different control is always like the sort of straight line, and then looks like the different treatments. You saw sort of a drop in abundance of these different species weights. Here's another one they had here, kind of showing these, I think, are environmental variables. And then this was, I think, just like a mock example of So weeks post-treatment, control against straight, and then the different treatments, and then the species weights here. So to summarize and conclude, canonical analysis is the merger of ordination techniques and multiple regression. Vector autoregression, extension of univariate regression with multiple time series, and PRC, principal response curves, extension of repeated measures, special kind, of course, of redundancy analysis. These are all really pretty advanced techniques that require certain types of data, careful construction, and really careful, concentrated, like nuance, pick apart, think around interpretation. So use these at your own peril, I would say. So with that, I would like to acknowledge the DaCCoTA project. Thank you for joining me through this 3-part series. Hopefully this was helpful to sort of get you a feel for multivariate analysis and what methods might be available for you and your projects. All right, thank you, and I will see you next time.